This last session for today, we want to talk about analyzing the Benthic data. And what are some different things that we want to look for in the Benthic data? How do we want to try to slice it in different ways? And what does it tell us about potential stressors? So again, going back to our analogy, where are we in this step in the process? This is really like conducting the autopsy, right? Looking at the Benthic data themselves and seeing, what did they die from? Interviewing witnesses, what are the ones that didn't die? What are they telling us? Who's there and present in the Benthic community? Who's not present in the Benthic community? And what can that tell us in terms of the stressors? All right, so I want to look at different ways to slice the Benthic data. The first is spatially. And so this is kind of why I emphasized before having multiple stations within a watershed. You can actually learn a lot about potential stressors just from what are the responses at different stations located in different places in your watershed. Okay, so in order to do this type of spatial analysis, we have to have multiple stations within the watershed. Okay, if we only have one station, then this is a line of evidence that we really can't investigate. Okay, so how can it help us? Well, it can identify potential stressors. It can potentially help us identify if there's some sub-watersheds that are of particular concern. Maybe it can even help us refine our assessment units and potentially delist segments. If we've got more Benthic stations scattered throughout the watershed, some of them are probably going to be unimpaired. And so maybe we can shorten impaired segments and reduce our number of impaired stream miles in Virginia, basically just by having more stations within the watershed and being able to differentiate better what's impaired and what's not impaired. All right, so now what can the spatial analysis tell us about potential stressors? Well, first we have to look at the network, right? And keep in mind that Benthic stations integrate everything from upstream. Second thing they do is we need to look at the magnitude of impairment, okay? The magnitude of the impairment, meaning how low is the Visky score, that should correlate with the strength of the stressor at that location, right? If we've identified the stressor as pH, as low pH, then if there's multiple stations, we should see a lower Visky score where we have lower pHs. Okay, and we can also, with different stations scattered throughout the watershed, we can look for upstream, downstream effects, particularly around a point source or something like that. All right, so let me use this particular example on the right to just kind of walk through some of the things we might be able to analyze from the spatial network of stations that we have. Okay, we start at station 5 here, and we see that we've got an impaired condition. That's a very small watershed. So what that tells us is within that very small watershed, the stressor must be present in that area. Okay, so that can maybe narrow down what the different types of stressors that we're looking for have to be located there. We added this within watershed reference, station 6. Okay, so we shouldn't see in station 6, we shouldn't see water quality parameters of concern that we see in station 5. And we can, in this case, compare what are the magnitudes of water quality parameters in station 5 versus at station 6. And in those places where we see differences, that should be an indicator to us that that's a potential stressor. All right, how about as we move farther downstream? Now, if we get to station 1, right? Station 1 is obviously integrating everything upstream. So it's integrating information from station 5 and it's integrating information from station 6. If we look at the scores at station 1 and the The Visky score there is kind of a weighted average of what we see at 6 and 5. Again, that tells us something. That tells us that the stressor we're looking for is still found in subwatershed 5. Now, if we look at station 2, now we have another impaired station here, which also means that the stressor has to be found within this subwatershed. And how does that stressor compare with the stressor at station 5? At station 5, do we have higher or lower Visky scores than at station 2? In this case, in this particular example, the scores at station 2 are even lower than they were at station 5. Okay, so now we can look at parameter data. Well, do we see, again, let's say a potential stressor that we're looking at is pH, low pH. Do we see lower pHs at station 2 than we do at station 5? If so, that's a line of evidence to suggest that yes, pH is a stressor, because the conditions here at station 2 are indicating that we've got more severe stress conditions here. As we continue moving on downstream, we look at, again, I just mentioned the magnitude of those stressors, but we can also look at, now as we move downstream to station 4 and then to station 3, what happens with those Visky scores. Okay, in this particular example, the score goes back up at station 4. Well, maybe that makes sense. What if we look at the kind of a flow weighted or an area weighted average of the upstream stations? Does that kind of predict what we see at 4? And if it does, then what that tells us is, yes, the source of the stressor is primarily from this sub-watershed here. Okay, and then as we move even farther downstream to station 3, again, in this case, it's showing conditions improved even more. Okay, again, kind of emphasizing to us that the source of the stress is in this subwatershed here, 2 and 5. It also, by seeing an improvement from 4 to 3, tells us that, you know what, this point source here is not really a source of stressor. The conditions have improved from upstream to downstream around that particular point source. All right, so kind of based on that spatial analysis that we looked at, which types of sources of stressors kind of fit the scenario that we see here? Well, the ag sources and the urban and suburban sources do. That's where we saw watershed land use wise, that's where we saw the highest intensity of those land uses. How about the point source? Well, no, not really, because we saw conditions improve from station 4 down to station 3, which bracketed that point source. So that point source is really not a considerable source of the stressor in this watershed. And then, is the stressor in this watershed something that's just diffuse? It's just kind of everywhere within this watershed? No, not really. We saw areas of the watershed that were unimpaired, and then we saw areas that were severely impaired. And as we moved down from there, we kind of saw a spatial average of those upstream sites, which tells us that the sources are coming from particular locations. So that's spatial analysis of the benthic data. It can tell us a lot. Another thing we want to look at is a temporal analysis. Okay, what's happening over time? And hopefully, again, here, we'll have enough data of these benthic stations being sampled time and time again, where we can hopefully develop what are some trends. Okay, are those trends going up? Are they going down? How do those trends relate to the water quality data that we've collected? Okay, if we see that both nutrient levels have gone up in the past 5 years, and we also see that Visky scores have gone down over that same time period, that might be an indicator. And then, are there other trends within the watershed? Right, land use trends. How has land use changed over time? Or specific events. Here's an example. The one on the right here was from a study we did in the North Fork Rivanna. There were 2 really low benthic scores on 2 different years. And when we talked to the technical advisory committee and folks within that watershed that were very knowledgeable, we found out that, well, you know what? In those particular years, there was construction on a dam, and a particular reservoir or lake was drained. There was exposed sediment for months on end, and a number of storm events during that time period that really flushed a lot of sediment out of this particular reservoir while they were doing construction on the dam and the outlet structure. Well, that kind of showed up in our Benthic data, and we saw really low Benthic scores at those times from what we know was a sediment stressor. So how does that relate to other times, perhaps? Maybe is sediment a stressor and it was more severe at those times and less severe at other times, but still severe enough to cause impairment? We talked about this before, I think, during one of the questions, and this is the seasonality of benthic data. Oftentimes, well, usually, our benthic data is collected in the spring and it's collected in the fall. And if we compare those spring and fall values, do we see significant differences? Again, on the right here, we see 2 examples. In one example, not really much difference between fall and spring. And then what's called the X-trib there, there's a pretty significant difference in VSCE scores between the spring and the fall. And in this case, spring scores were much lower. So what does that tell us? Well, things like sediment and nutrients, as I said before, they are often in higher loads and higher concentrations during spring runoff. Okay, so we may expect spring scores to be lower if those are some of the sources of impairment or sources of stress. Kind of the opposite, if we have conditions where fall scores are lower, perhaps temperature and dissolved oxygen could be some stressors that we want to take a closer look at, because those are particular stressors that they're going to have their worst conditions in the late summer, early fall time frame. And so that would have a more direct and more immediate impact on fall benthic sampling results. Now we can also look at individual metrics. Okay, the VSCE score is a multi-metric indicator. It uses individual metrics that are combined and averaged to come up with the total score. But there's information to be found within those individual metrics as well. And they may tell you something about stressors. I think Nesha shared a particular figure that looks somewhat like this, where we were looking at both a reference station within the watershed as well as the impaired station within the watershed to see which of the individual metrics really changed, which ones were driving the low VSCE score. And those different metrics can sometimes tell us different things. That first grouping there in the green, those could kind of indicate almost any stressor. Those are really measures of species richness and sensitive species. And so those are the species that have those really narrow tolerance ranges. And if we begin to move the environmental condition in one direction or another for any particular parameter, we're going to begin to lose those sensitive species. And as we lose those sensitive species, we're also going to lose richness, which is just the count or the number of different species that we have. And so those things are going to go down. Sensitive species and species richness, they're going to go down with any stressor. So that grouping doesn't necessarily tell us a lot about what a particular stressor may be. The next grouping there, scrapers and chironomids, those could potentially be indicators of sediment stressors. If we have really low scraper abundance, that could be an indication that sediment is covering up benthic substrate. Scrapers aren't able to cleanly scrape periphyton growth from the tops of rocks because maybe that's getting smothered by excess sediment. And then the chironomids, those are a species that really like to dominate in in sediment-rich conditions. And so if their numbers are very high, that may again be an indicator of potential sediment stressors. And then the last two, %2 dominant and the biotic indicator there, those are both kind of more indicators and metrics of nutrient or organic enrichment. And so we see if those are really driving the low VSCI scores, that maybe nutrients, organic matter, could be some of the stressors that we want to take a deeper look at. What's the reason for that? Well, if we have a condition where we have lots of food, basically, nutrients and organic enrichment basically means a lot of food, we're going to have very tolerant organisms that can outcompete others. They're going to dominate. There's plenty of food for them, and so survival of the fittest, whoever can outcompete is going to dominate in terms of percentage of the community. So that's usually conditions that you may see where you have excess food, basically. So that's a way that you can kind of look at the individual metrics. Maybe they tell us something about particular stressors, maybe they don't. Another thing to look at is the composition of the community. And you can look at this by order, and maybe some orders you might want to break down, particularly Trichoptera, you might want to break it down into your Hydrocycid versus your other Trichoptera. But the pie chart is a really good way to look at this different composition of the community, and you really need to compare it to some reference. In this case, this small stream reference that's shown here, I mean, just a quick glance at the pie chart tells you, pretty balanced community, right? A lot of different colors, and then the representation of those different colors, which are different benthic macroinvertebrate orders, you know, they're somewhat evenly distributed. If we take a look at this X-trib to flat branch here, again, we see a different picture. The community is dominated, in this case, by Diptera. I think in this particular case, it was Chironomids that were really dominating the system. There's a lot of other here too in this particular system. The gray here was other, and in that case, it was mostly oligocete worms. So here we've got a system that's dominated almost 50% by Chironomids and another 20–30% by oligocete worms. Well, what are the conditions that those organisms like? Those organisms like sediment-rich conditions. And so that gives us an indication that sediment may be a particular stressor in this stream. Okay, this light yellow here is the Hydrocycidea. Well, if those are a large portion of the community, that might tell us that nutrient enrichment could be a particular stressor in this watershed, because those hydrocycids, they're net spinning caddisflies. They will often be present in high numbers when there's a lot of food, particularly when there's a lot of food that's flowing in the water column. Okay, because they're going to catch that on the nets that they build and then get their food from there. So what does the distribution look like? And then what can we find out about that distribution in terms of what are the preferences for those organisms that kind of dominate the community? And then functional feeding group. Again, Nesha showed us an example of this type of figure. It's a stacked area graph. I think it's really useful for looking at functional feeding groups. In this case, what we're looking at is on the left-hand side, a reference, and then on the right-hand side, our impaired site. And by using the stacked area graph, we can really see where there's changes in the feeding strategies of organisms. In this particular case, what we can see is this blue area are collectors. And as we move from the reference condition to the impaired condition, those collectors grow in a percentage of the community. Okay. We also see in this same case that filterers, represented by the red here, as we move from a reference condition to the impaired condition, those filterers grow in their representation in the community as well. So what do those two things tell us? Well, how do collectors and how do filterers get their food? Well, the collectors rummage around in the sediment, collecting things to eat. The filters, they're filtering things out of the water column. This could be an indication of a sediment stressor. Okay. And then what do we have happening up here? Well, we've got shredders, they're decreasing. Scrapers are decreasing. Okay. What might that tell us? Well, that could be a again, a situation where sediment could be covering up substrate on rocks so that the scrapers and the shredders don't have an available food source, so they're going to decrease in numbers. And then those things that rummage around in the sediment looking for food, like collectors, are going to be increasing. How about if we have a nutrient stressor? Well, we're going to find that maybe these scrapers increase. Okay, so if the scrapers are increasing, what does that tell us? Well, that means that there's more food available of algae and biofilms growing on rocks. That's going to lead to an increase in the scraper population. So again, maybe an indicator of nutrient stress. And then total abundance is usually going to increase when we have nutrient stressors as well. We can't always get at an exact abundance from the benthic data that we collect, but there are some indicators in the benthic data that give us an idea as to what those abundance might be. So the homework that I had for you today that you'll see under handouts in the section of the webinar was to make one of these area charts. Now, Nesha, tell me, did I hear in your presentation that this is generated for you automatically from that query tool?
No, that's just that data can set up nicely to generate one of these, but that would be cool.
Oh. Good. Okay, I was going to say, well, then don't worry about this practice if the tool does it for you automatically. But if for homework you want to look at there's two files there. One's an Excel file which has some artificial benthic data in it, and then there's a Word file. And what that has in it is some instructions for walking you through the steps that you would take in order to take a typical output of benthic data and then turn it into this type of functional feeding group area chart. Okay, so that's something you might want to do for homework, and we can look at the results on Wednesday as we open up. Another piece of information that's just been recently available to us is the biological condition gradient data. I think it was some of our biologists at DEQ as well as others maybe in other states. I'm not sure exactly how it was developed. Really sat down and went through a lot of different taxa and scored those different taxa based on what their tolerances are to individual pollutants or to individual parameters. And so it's basically kind of going back to what we talked about before and looking at those curves, okay? And then looking at those curves for different parameters. And then for a given species or a taxonomic group, for a given taxonomic group and for a given parameter, basically how narrow is that tolerance curve? Okay, and so basically looking at all of that information for a lot of parameters and for a lot of different taxa, they were able to score and come up with what we call the biological condition gradient score. It looked at 560 different taxa, 10 different stressors. One of the pieces of information from this that can help us the most in the stressor analysis is attribute 6. Okay, and that is tolerant taxa. Which of the taxa are most tolerant to these particular 10 stressors that were looked at there? And for this attribute, a score of 5 indicates that that taxa is very tolerant. So what that means is that if that particular taxa is present in high numbers, okay, then that could be an indicator that that particular stressor is controlling the community structure in this particular case, and could be a probable stressor. All right, so let me show you a couple ways we can look at the biological condition gradient data. One of the first ways is look at the predominant taxa within your stream. I suggest maybe the top three. So which three taxa are most prevalent within samples from the stream you're looking at? And so here's an example here. 3 different streams, Bailey Creek, Nut Creek, Nut Tree Branch, Rowan Creek, and here are the top three for each, top three predominant taxa. And if we look across the different parameters here, we can see, where do we see scores of 5? Okay, those scores of 5 tell us that that particular taxa is going to be likely in high numbers under the conditions of this stressor, because they're very tolerant to it. Whereas other species that are less tolerant, they're going to decrease in their prevalence. Okay, so in this case, what do we see? Well, some species like the oligochaetes, they don't really narrow it down for us a whole lot, right? They're predominant under a number of different stress conditions. Okay, so that doesn't help us narrow it down much. But here's a case. The simulium, it's a type of black fly larvae. We see that they are highly prevalent in this particular stream. And in this case, the biological condition gradient tells us that they're going to be in high numbers in places where there's nutrient as a stress and maybe sulfate as a stress. Okay, so this might be able to tell us something about what some of the primary stressors are in that particular stream. We also see here, look, here's another, the Canis. That particular taxonomic group, it's going to be present in scores that are correlated with high imperviousness and relative bed stability, low log relative bed stability measures. So that's an indicator of sediment. Okay, so this particular species appearing in large numbers at this site might be an indication that sediment is a stressor. Okay, and we see it here. We also see it in this Nut Tree Branch station. Okay. So what's another way to look at this biological condition gradient data? Another way to look at it, so we looked at what are the most prevalent species, but now let's look across all species that are present there, okay? And let's take an average, and you could do this as a weighted average based on community composition, or just a straight average of what's the average attribute 5 score for across the different taxa, okay? And average that by each of the parameters. Okay, so that's what was done here. And then once we had that average score data, then we ranked them based on from lowest to highest average score. And so what you see in this particular table are those ranks. Okay, so what this means is numbers of 1, 2, and 3, those were the top 1, 2, or 3 stressors that had the highest average biological condition gradient scores across all taxa at that station. Okay, so what does this tell us? Well, this is really great, because now if we look across these different stressors, you know, here's a couple of stressors, they're probably not likely going to be stressors. They ranked as the lowest average, okay, across all taxa for those stations. Some things in the middle, but here's numbers 1, 2, and 3. Okay, so for each of these stations, each of these streams, things having to do with nutrients and sediment were either 1, 2, or 3 in terms of the highest average biological condition score across all taxa for that station and for these particular stressors. So kind of a good indication that those might be probable stressors in these particular streams. All right, it's our last time to check in. If you have questions for us, go ahead and drop those in the question box. Again, here's some individual things to think about that might spark ideas for future discussions as well. Maybe some guidance on biological condition gradient data, how to use that. And then how are you utilizing your Benthic data? And are you utilizing all of it to be successful in the Benthic stressor analysis? So let me pause here. I'll let Rob and others chime in with questions as they come about.
We've got one comment here, Robert. William says that DQ Benthic data does have number of grids to reach target count recorded. This can be used as an indicator of total abundance.
Exactly. Yeah, we don't exactly have an abundance number, but you're right. Number of grids that you needed to get to the target number of organisms can be an example of, not example, but can be an indicator of the number of abundance of organisms within that sample. And that's actually a really good indicator, particularly for nutrient stressors, because you're increasing the food, you're going to increase the population levels within that stream. So yeah, I would encourage using that type of information in your stressor analysis as well. If you have a case where, maybe you had to subsample 10, 15 different quadrants in order to get your number, that's pretty low abundance in that area. And if you have, along with that, if you have very low VSCI scores, that might indicate some type of toxic, meaning that something toxic is killing organisms at the site to keep your abundance very low.
First, Robert, thank you for the session this morning. It has been really, really good and very informative, so appreciate that.
Good. Appreciate that.
Oh yeah, this is very helpful. So I guess the first question is, in general, about reference, the use of references and kind of the conversation as it evolved. It seemed like the use of the reference, at least at this stage, was more qualitative, if you will. I mean, I know there are pie charts and different types of analysis and kind of comparisons of our impaired sites to a reference site or sites, but just kind of getting a sense for how it's lining up that the reference or references will be used, and related to that, in this particular case, we had one reference site, and I guess ideally a reference in the same watershed would be great, but if not, it looks like we have one reference, and the use of one versus more than one, and just any thoughts or comments on that, and if references are more qualitative in nature versus kind of specifically quantitative in setting endpoints, maybe the number of references isn't as potentially significant. If that makes sense. There's a couple questions in there.
Yeah, so I think first of all, you're right, a reference within the TMDL watershed is best. I would say that the use of the reference data is quantitative as well, because you can statistically compare any parameter across the reference versus the impaired watershed. And so if you see a parameter that statistically it's much higher in this impaired watershed than it is in the reference watershed, well, that's one indication that that particular parameter could be a stressor. Now, it's again not a smoking gun, which is why we use multiple lines of evidence, but it does give an indication. Now, on the flip side, if something is basically not different some parameters not any different between your reference and your TMDL watershed or TMDL site, that is a smoking gun to basically say that stressor is, or that parameter is not a stressor in this particular watershed. And so it can actually be used, quantitative comparison between the reference and the TMDL watershed can almost be used more strongly to reject stressors if we don't see differences in that particular stressor, the different watersheds. On the flip side, it's not a smoking gun, but it could be a line of evidence to suggest what stressors are. But in terms of the benthic stressor analysis, in neither of those cases are we really setting thresholds based on the reference. Did that answer kind of help?
Yeah, it does. And I guess part of the conversation related to that, one aspect is a single reference site versus multiple reference sites and any preference on that. And I guess related a different issue, but related to the conversation of stressor or references, is how and I think it comes back to the weight of evidence that you just mentioned, but how does use of a reference or references, one or multiple sites, how does that complement or tie into the thresholds that are built into the tool and kind of give you your stoplight colors and outputs? Because I know that's something that we staff will certainly, the first kind of run through things will be the output of the tool, and it seems like maybe the reference sites is building upon that.
Yeah, or actually the other way around. I think the benthic stressor tool is kind of building on the reference site idea. So where the data came from for the benthic stressor tool came from all the ProbMon data. So there are a lot of reference sites represented within that data set. And so, when you're using the benthic tool, it's basically accounting for all those multiple references, but it's also accounting for multiple impaired sites as well. And so you kind of have both worlds, right? You have comparison across reference sites, but you also have comparisons across other impaired sites. And the tool's really, developing correlations between that data set and a particular parameter, and which is where those thresholds come from. What levels of different parameters are associated with impairment at, I don't know how many sites are used, but there's hundreds of sites, I'm sure, across the state. So I think, with the addition of that, I don't think there's really a need for to look specifically at multiple references. I think one reference that is as close as possible to your TMDL site, I think that gives you a valuable piece of information, and that piece is different from what you get from the benthic stressor tool. You don't have the same regional location, you don't have the same maybe local factors that you have with a reference. You don't necessarily have a stressor tool because you're looking statewide. But you've got other advantages because you've got a larger data set, you've got more references, and you also have impaired sites that are. So I think it's two different tools, two different lines of evidence that both have value.
Yeah.
And I think too, Robert, with using that Caddis model, that really allows you to integrate the results of that stressor tool and the results of your reference comparison, where you can weigh all of those factors and, use both to support or refute your different stressors.
Exactly. Yeah. So for example, the Benthic stressor tool may say, oh, wow, chloride levels were really high, and the chloride levels are in the, high probability range for stress effects. But then if you have a very local reference that's unimpaired and you have the same high chloride concentrations, that would refute that, right? And comparing those two lines of evidence, they say, well, I don't think chloride is a probable stressor in this case. Because look, I've got a local situation where I also have high chlorides and I don't have impaired benthic conditions. Maybe that's just a local condition here, in this region, which gets back to some of the things we've dealt with in Sand Branch, right? The underlying geology, Triassic Basin.
And Robert, I think we've talked about this, but I thought I'd throw it out for thoughts or comments while we're, in this session. And it has to do with the discussion on the BCG, and I don't have it in front of me, but the last column that identifies percent impervious. I've noted it before, but it just, that particular variable or parameter, it's a little different. The others generally are, like a physical or chemical water quality related, and many of them are pollutants or 0.2 pollutants, whereas percent impervious is more of a land use condition. And while I understand the tie-in of imperviousness as it relates to degraded water quality in certain benthic community, it just kind of, to me, when I look at that table, it stands out as a little bit out of place, and it's not necessarily something we're going to address through the TMDL program. And I know the benthic stressor analysis is pointing at all the stressors, whether they're pollutants or not. But just, I think in the context of that table, it always just kind of stands out to me. So any thoughts or comments on that?
Yeah, I would agree with you there. Maybe somebody that was involved in the development of the biological condition gradient could speak better to that as to why that was chosen as one of the parameters to look at there. I don't know for sure. My guess is that they were looking for something having to do with sediment, and there are not great sediment measures, and that was a kind of a surrogate. But I don't know that for sure. So I would probably, want to, want to get an answer, a definitive answer from some folks that were actually involved in the development of the BCG.
No, that sounds good. Thank you.